Okay, we are live now. So good morning and welcome everyone to the first lecture in PNS Project and Seminars Mobile Genomics course. And for today, we will discuss the introduction and the project proposal. However, for the project proposals will be a private session. We are going to turn off the live streaming and the recording because these projects are ongoing projects normally. But yeah, I will be happy to discuss any project ideas, all of us basically, with any of you who are watching us on YouTube. If you have something to discuss with us, or you would like to join us, if you are basically among the top 2% in your class, so you can contact us to join our group basically, or to do research remotely. So I'm Mohammed Alser, and I'm lecturer at ETH Zürich. And let's get started to the course. So what is the role of this course? So as you know, this is a projects and seminars. So we will cover the basics of genome analysis to understand the speed accuracy trade-off in using computationally lightweight heuristics versus accurate computationally expensive algorithms. So most of these genomics applications that we are targeting normally use really computationally expensive algorithms to do rigorous analysis. But sometimes we don't really need this rigorousness to get the exact same results, especially when we are talking about a huge data such as genomics. And there are a lot of heuristics already proposed in the field, but some of these heuristics are not well characterized, for example, so we don't know their drawbacks, their advantages, and so on. So we are going to use some of these in some of the projects, but some other projects are totally novel, proposing totally new algorithm. So it depends on the student choice to select one of these projects. Student will basically do experiment by themselves to evaluate the different heuristics algorithm and observe their effect on the end results. So again, we don't know the characteristics of these heuristics, whether they are new, proposed by us or by someone else. We might see a new application that even the author of the algorithm was not thinking about in that time. So we might evaluate these algorithms on a new applications, basically. And our end results, or eventually, we will evaluate these algorithms, and this evaluation will basically give the student the chance to carry out a hands-on projects, to implement one or more of these heuristic algorithms in their smartphones or portable devices. And that hopefully will help the society by enabling on-site analysis of genomic data. Why we need on-site? Because think about the pandemic, for example, where we have really a population need to analyze their genetic material or do PCR test or do any kind of analysis regarding the genomic material. So if you'd like to use a center or centralized computation to handle all these population scale analyses, then it will take months to do so. So it will be very nice to have some portable devices where you do near patient analysis, for example, or just out of curiosity where you pick a sample from the environment and try to analyze it on site. This is also true, for example, in space. So think about the space setup, where you send a very small robot to take a sample. So you don't want to wait until you get back to the Earth to do the analysis, but rather it will be nice to do all these analyses within the robot setup itself. And that imposes restrictions on the weight, on a lot of things, temperature, and so on. So this shows the importance of having really portable or mobile genomics. And this is the goal of the course. So what are the key objectives we have? So basically we target multiple components that aims at improving the student first basic knowledge. So as a prerequisites, we don't need you to have any basic knowledge in genomics, for example. So we are going to teach you. We have very great teaching stuff they're already attending today. So they are going to teach you on different topics about genome analysis and the things you need basically in each of your projects. And that will provide you technical skills in genome analysis, how to do the analysis by yourself, as well as computer architecture, because you are going to implement algorithms. Sometimes you may pick a GPU projects or FPGA projects and so on. So you will also get some hands-on experience with these projects. Since we are doing sometimes new algorithms, that will also provide you critical thinking and analysis. So once you face a problem with any of these projects, so we will teach you how to tackle that problem, how to basically attack the problem, such that you do research first on Google, where you can search for similar problems or error code. And sometimes you find it in the research papers. They mention this problem and that problem, and they propose some method for the solution. Then as a critical thinker, you need to try this solution and get your own observation when each of the method fails, when each of the method is doing great, and so on. So this is how we do research, even ourselves. And you'll hopefully, by the end of the course, you will be familiar with research directions that we are proposing to you, plus how to do research. So the main goal of this course is basically not to do research, since it's seminars or projects and seminars where typically just need to learn something new, but not to do research. But we also help those whoever highly motivated students that would like to do something new or carry on projects that take longer than the semester, or they want to take over another project after finishing the course and so on. So we have all these available options after completing the course. And toward the end of the course, you basically need to present your progress or the entire project to our research group. So we are about 40 members. So these will be attending your presentation at the end of the course. But it will be in a funny environment, a very friendly environment. So don't take it seriously, but you need, if you do really well within the semester, so I think the presentation should be just a matter of formality. All right, so what's the key goal of the course? Basically, you need to learn how to efficiently implement one of the key steps in genome analysis on portable devices. Again, genome analysis is not a single step, it's not simple computation, but they have a lot of applications, a lot of computation going on, and many steps. And it's really hard to learn everything within this semester. So basically, you will pick just very small step and try to improve it and try to do a thorough analysis on portable devices. Again, what are the prerequisites of the course? As I said, no prior knowledge in bioinformatics or genome analysis is required. We will provide everything, basically. We'll provide teaching over the next weeks. So we have, I think, about 11 weeks remaining from the semester. So every week you will get about 30 minutes of teaching, just try to teach you about different topics related to what we will have in terms of projects or in terms of the course. And most of the projects, we require really good knowledge in C programming, and that is, I would say, a must. So without using C, you may not get efficient implementation. You could try Python for those new ideas that need a proof of concept, just to show that's working fine. But I hope every one of you already got a really good knowledge in C programming. You need to show really interest in making things efficient and solving problems. Don't get fed up once you face a problem. You need really, as I mentioned, to tackle it. You need to find many resources trying to find the solution. But also at the same time, we have weekly meeting where all of us as a mentors, as a teaching staff, will be meeting with you. So whenever you face a problem, you can also discuss it with us and consider us as a valuable resource. So try to leverage that resource and ask many questions as possible, attend these meetings and show your progress on a week basis. So this course is provided by Professor Onur Mutlu, who is a professor at ETH since September 2015. He was also professor at CMU, Carnegie Mellon University, for about 7 years. He got his PhD from UT Austin, and then he worked at many companies where he established computer architecture research group at some of them. And the best way to reach him is his Gmail account. And as Safari Research Group, we are interested in different research directions, including computer architecture and bioinformatics for sure. I'm also leading this course. I'm Mohammed Alser again. I'm lecturer, senior researcher at Safari Research Group. I got my PhD from Bilkent University, Turkey, where my PhD thesis was tackling the acceleration of genome analysis, and was awarded the IEEE Turkey Doctoral Dissertation Award. And I like the best way for communication, or at least sharing the research about bioinformatics, is Twitter. My main research, as you can guess, is bioinformatics, computational genomics, metagenomics, and computer architecture. We have nice teaching staff, mentors for projects. So we have Juan, who's senior researcher and lecturer. We have also Noor. She's a senior researcher with us at Safari. We have John Fortuna, PhD student. We have also Max, who was taking a seminar in computer architecture within, before a year and a half, almost 2 years, I guess. And he did a really good job within the seminar course. And then he decided to join us as a researcher to do research and continue what he was starting during the seminar course. And he's doing really good job with the research until now. And I hope he's enjoying. So he will be among the teaching staff. Hopefully he will provide you a good help as he's still a master's student. So you can take closer experience from him as well. All right, what are the requirements as a student in this course? First, attendance is really required for all meetings. As I said, there are about 30 minutes to 1 hour of teaching, and then another 1 hour of meeting with mentors every week. The teaching, I would say it's up to you to attend it, but I really recommend everyone to attend this lecture. They will be recorded. They will be hopefully live streamed on YouTube, so please attend them. You will get more experience about this topic. It will be important for your growth. But attending the meeting for progress is really a must. Without these, I think you're not going to do or maintain a good progress throughout the semester. You need basically to study the learning materials, especially the one we assigned in the first week. And also, as you go with the progress, we're going to assign more learning materials that are deeply related to the project you are handling. So like when you face a problem or when you need a new idea to tackle something, we are going to assign a paper just to read the method, get more ideas about how to solve it and so on. So as I said, as a student in this course, you need to handle only a single project. Basically, you will build, implement, code, and sometimes design with a close engagement from the supervisors. And you need to participate in this meeting, in all other meetings. Please ask questions, contribute thoughts, ideas, anything you may have a concern. Read relevant papers, basically, if we assign them or without assigning them, just if you find something interesting, share it with us, and then we can guide you as well. You'll be responsible for preparing a presentation at the end of the course and uploading all the material you did within the semester to a GitHub repo that we will create. The course website is already ready. You can access it. We will create a Moodle soon after the meeting, and we will share the project topics over there on Moodle. You need to rank them with the most favored one and then least favored one in the project that you don't really want at all. And then based on this ranking, we're going to assign the project to you. Then we give you another week as a chance to discuss the project, get more details, and then you may want to change your mind, and then we confirm the selection. And basically, you can always contact any of us over Moodle directly, so all the students will share the same concern or will know the answer, so they don't need to ask the same question again, or you can directly contact us by email. Feel free to share any things on Moodle, like learning material that be benefit for everyone and so on. So we will have next meeting as well. we will decide the time by email. We'll see the best time and then stick to that time all weeks. So hopefully we will come to find a single time slot that is good for everyone. It's really important that you study the learning materials again, which especially the things we assign in the first week. And we will start the projects basically next week. Hopefully today we will send you the choices for the project, and we will explain them right now, all the projects, then you have probably a few days to select the project. Okay, so that was all for the introduction of the course. I don't know if you have any questions, and we can take them now. Is everything clear? Okay, I see a sign of yes. All right, so now just a brief introduction to give more time for the project discussion. So what is genome analysis? Okay, as you can see in this picture, there are a large number of individuals. They are really different in phenotypes. You can see skin color, hair color, the length, and so on. So all of these are caused by some genetic variations. And to do analysis to the genomic material, you would like really to identify, measure, or compare genomic features, or even functional element annotation at genomic scale. Why is that important? I give you already a hint. It could be important for understanding genetic variation that cause phenotypes and disease, for example. So phenotypes not necessarily to be harmful disease, but also you can use it for predicting the presence and relative abundance of microbes in a sample. That's what we call metagenomic study. So in metagenomic, you just swap something from crowded places, and you don't expect to have single genome, but rather you have large number of viruses, bacteria, traces of a human, traces of fruit, and so on. So it's really hard problem, different from genetic variation. Genetic variation is still hard problem again, especially for the disease that we don't know the causes for. And but all of them at the end use some kind of genome analysis. You can still use genome analysis for rapid surveillance of pandemic, as we have in COVID-19, for example. You might have curiosity to understand how the COVID-19 move through sewage water or the air-conditioned ports and so on. So all these kind of questions can be answered by doing some kind of genome analysis. One important aspect also or application is personalized medicine. So when you go to a hospital, sometimes when the symptoms are not clear, so the hospital will do genetic test for you, and based on the report, they will match it with the previous patient that they already have historical data about, and they know that the previous patient get well because they got this medication. So probably they will prescribe the same medication to you as well. Hopefully, since you have the same genetic material, you will get better as well. Okay, so hopefully by that I already highlight the importance of doing genome analysis, and now how to do the genome analysis. So assume you have a genetic material. The end goal is to get this complete sequence of genome, or the DNA, which is just basically ACGT letter or alphabets. However, we don't have any machine that give you this as an answer. What we have is first, you buy these commercial kit from the pharmacy or online or from store. You take a swab from your body yourself, and then you send back the swab to the company again, and the company will use one of these machines. These machines can be really huge in size, like fridge size, or it can be handheld as we have in MinION devices, which is portable. But all these machines, they have one thing in common, which is they produce reads. These are subsequences of your DNA, but not the complete sequence of DNA. And all these reads still lack information about their order and location. So we don't know really which chromosome these reads coming from, and how they look like in the original DNA, whether the first one is first or the second one is first, and so on. So it's really generated at random level from the reference genome or from your DNA. So you can think about it as the puzzle solving analogy, where you have the reference picture, you have the pieces, which are the reads coming from the sequencing machine, and then hopefully you can match them to the reference picture and stick them together. So you try to solve them, and this will be your DNA, and that is what we call a reference genome that is already built for the entire human population. So the reference genome is already publicly available. It looks like something like this. We call it FASTA file. So the first line is unique identifier to just represent the human or the target individual. And the followed line are basically the sequence itself, or the DNA complete sequence. And it's publicly available. You can download it yourself. As I said, we have only single reference human genome that is representative. But again, there are a lot of problems with using just single for all population, which you can think about it as a bias, because we don't expect African to be similar to European, to American, to Asian, and so on. So it will be nice to have a reference for each population, but given that this reference genome needs 13 years to be generated, plus some other 15 following years to complete it or improve it, then that is a huge effort. So it's really hard to build an accurate and complete reference genome. The read set coming from the sequencing machine, we call it FASTQ file. And we have 4 lines per read, per sequence coming from the sequencing machine. We have 4 lines. So the first line is unique identifier, as in FASTA file. The second line is the read itself. You can see it's ACGT. When we have ends, mean the sequencing machine cannot read that base due to complexity, for example, in the DNA itself, because you know the DNA is coiled in your cell, so it's really complex to read. The third line is just separator plus sign. The fourth line is the quality of, or the confidence of the sequencing machine when it was generating C. So each character is the confidence of the corresponding character over there, and that is in ASCII code. The read set is also publicly available. This is the nice of sharing your genomic material or the data to enable or empower research in this field. So we can access a huge amount of genomic data online. This is one of them. You can see information about the machine was used to generate it and many more data. Okay, so now we have the genetic material. We send it to the sequencing machine. We got reads, and we have the reference genome. So we take one read, try to align it to the reference genome. Until we align all these pieces, we find the right position for each piece, and then we build your DNA back. So how to do that in practice? We have the FASTA file, we have the FASTQ file. So the FASTA file is the reference. So we take pieces, it could be randomly or it could be using sophisticated method. There are a lot of methods how we pick these pieces. But anyway, we pick these pieces and we store them in something like indexing data structure. As you know, indexing data structure is in the query process. So you can quickly search for any piece of data inside this file. We store them, and then we jump to the FASTQ file, which is the read set. As I said, the second line is the read. So we go to the second line, we collect pieces. It should be the same method we use for collecting the pieces from the FASTA file. And then we query the blue piece. It exists here and over there. Then we query the green piece. It exists here and there. And then the red piece. And then we merge all these locations. Whenever each of them exists, we merge all this list of location and try to make sense out of this location. So for example, this blue piece is already followed by a green one and followed by a red one, right? So it makes sense to pick something in the reference genome that has exact same order here, which is, as you can guess, this line. So you can see the blue followed by green followed by red. And then we do base by base comparison. So we take that piece from reference genome and we compare it to the read. We do base by base comparison using what we call dynamic programming algorithm or edit distance algorithm, if you are aware about these kind of algorithms. What's the goal of doing that? So as you know, we are looking for genetic variation that cause a disease or a phenotype. So we really need to know what's the location of these variation and what type of variation we have. It could be insertion, deletion, substitution, and many more. So you really want to know the exact location of these in your DNA and what type of differences. And for that, you really need to use dynamic programming algorithm. When we do the analysis for this entire process, we find the sequencing machine really capable, it's really fast, can handle 1 human in 1 hour. But when we do the analysis, it's still bottlenecked in read mapping, assuming the base calling, which is another step, it's done in parallel with the sequencing. So that was for genome analysis. Now, how about intelligent genome analysis? So I was showing you the bottleneck, the step, how you do it, how to improve the efficiency using indexing scheme, some filtering, and so on. But speed is not the only important metrics here, since we are dealing with genetic data, which can be think as medical data. So fast genome analysis is one important metric, but also we want to use energy efficient devices to do the analysis, given that we are targeting population scale. Plus the scalability. So you want to scale this. It's not about how fast you can do one analysis, but how many analyses you can do in a given day, for example. That is the scalability. So it's very important to meet this metric. Also, the DNA is a valuable asset, so we need to worry about the privacy. We don't want to share the genetic material with everyone, because based on your own DNA, if you are willing to share it, I can infer the DNA of your brothers, your sister, your parents, and so on. And that's very risky, especially for health insurance. If they know you have this disease in advance, they may block you from getting insurance and so on. But don't worry, there are a lot of methods to share the portion that you sign a consent for it. So for example, if you are going to test against a certain disease, there is no need to share the entire genomic material. You can do the test for only that portion where you know that there is a genetic variation may cause that disease. So you can share some pieces. Or there are a lot of research that, for example, Noor going to explain how to hide some of these data or impose a noise on them that may not affect the interpretability of the data, but it will hide the relationship with your brothers and sisters and so on. So there are a lot of solutions. So I still encourage everyone to share their genomic material for research purposes, given that the platform is really good for that. The research is ambitious and so on. We really want to maintain accuracy, so we don't want to hurt the accuracy just because we are going for faster genome analysis. Doesn't mean we want to give wrong results, because this is very risky to tell a patient you have this disease, but actually they do not. So we want basically to have fast genome analysis in about seconds. And we want to do this using limited computational resources. As I said, it's really important to do portable devices rather than using cloud or clusters or big machines to do the analysis. I think most of you watched these 2 movies, so you can get an idea how portable devices look like. Can be smartwatch or anything basically can do the genomic material or the analysis really quick. Okay. So with that given, so we don't know where to do the analysis. So these days we are doing it inside the CPU, but you could also do it in FPGA, GPUs, in specialized devices, in ASICs, or inside the main memory, for example. If you take the courses from Onur Mutlu, you must be watch a lot of lectures about in-memory processing, near memory processing, or near storage, for example. Or you could enable the analysis inside the sequencing machine. That is still open research question. But at the end, we would like to have something like this. This is a real device, a real sequencing device attached to an iPhone. The accuracy of this device, as you can guess, is not that high. Also, the throughput is not that high since it's a small device. But at the end, we hope the technology or the sequencing technology get improved such that we will be ready for this if we prepare portable analysis plus the sequencing. Then we will have a complete system, can do the analysis in portable fashion, really quick and affordable way. As I said, the privacy is very important. You can read this paper if you're interested. Knowing more about the attacks can happen to your genomic material. And yeah, why it's important to do the processes in different ways than what we have in traditional way today. So basically data movement. We have a huge amount of data moved from the sequencing machine to the storage, to the main memory, to caches, all the way to the CPU to do the process. And just accessing the main memory to fetch a piece of data is really more expensive than doing a process or a complex process in the CPU. So you can see 160x at least more energy than doing complex addition operation inside the CPU. And that makes data movement really dominates performance. So it's important to solve this data movement because it accounts for 40% of the total system energy. We are not talking about computation now, just data movement, just about 40%. And that is a huge, especially if you are talking about portable devices. The first goal in this course to do efficient algorithm. By doing efficient algorithm, then the second goal is to implement these in hardware, piece of hardware. It could be GPU, FPGA, or it could be customized chip and so on. Again, this is the same question, where to place the processing. And for that, we have an open question, how and where to enable fast, accurate, cheap, privacy preserving, and not only that, but also want it at the exabyte scale or population scale analysis. But remember, you can improve the algorithmic level, or you do algorithmic optimization. You can also do novel architectures for hardware. But if you do both of them, then you will gain the best speed up that you can achieve. And we've already proved this in many of our work. You can read them. I'm assigning them here. If you are curious, you can watch our lectures or read these papers. Especially this can give you an overview about the entire read mapping process and many other applications that use genome analysis. This is another paper showing you all the steps, all the algorithms are currently used. And this is the same paper. This is a video lecture you can watch. That is, I think, a talk I give at ReComp this year, which is one of the best bioinformatics conferences. This is another lecture. It's really long. It can give you all details. It's about 3 hours lecture. This is a lecture by Professor Onur Mutlu, 1 hour and a half, and these many more papers from our research group and from many of the teaching staff that are attending today. So that was all for the introduction. I hope you get something useful for today. So if you have questions, I can take them now. I don't know if we have questions on YouTube. If not, we can jump directly into the project proposals. But as I said, that will be a private session. So we are going to turn off the live streaming. All right, that was great. Let me stop sharing. And